today I've got the absolute pleasure of presenting the CARD NIH initiative today. So before I jump into the data and the methods that I'm going to present, I'm just going to give you a little bit of background on what CARD is and a little bit of background on the genetics of Alzheimer's disease and related dementias. So CARD is based in America at the NIH, and it's the new Center for Alzheimer's disease and related dementias. And CARD's central mission is to initiate, stimulate, accelerate, and support research that will lead to the development of improved treatments and preventions for these diseases. And we can use genetics as a starting point to identify treatments. So if we identify the genetics, then this gives us a better understanding of the biology, and then we can identify genes to target with drug therapy. But what we've realized in the last decade is it's not just enough to understand the biology and to identify a target, but we need to dissect the disease. And what I mean by that is we need to be able to identify patients way before they show signs or symptoms. And we also need to be able to divide the disease into mechanistic subtypes, all moving towards identifying the right patient at the right time to apply the right treatment. And so just a little bit of a background about the genetics of what Alzheimer's disease and related dementias. So Alzheimer's disease, or AD, is the most common type of dementia and has a very clear genetic component. Highly damaging variants in genes such as APP, PSEN1, and PSEN2 have been identified as disease-causing. And the most recent GWAS identified over 75 risk loci. So Lewy body dementia, or LBD, so this is the second common most form of dementia, and it's likely on a genetic and pathological continuum between Parkinson's disease and Alzheimer's disease. And then finally, frontotemporal dementia, or FTD. So cases report a positive history in almost half of cases, and several important genes have been included, such as MAPT, GRN, and the C9orf repeat expansion. And so we know that these diseases have a very big genetic component, and we know that they've already been associated with regions in the genome that are repetitive and structural variant rich. However, all of the previous sequencing efforts for these diseases have focused on short-read sequencing, and we know that that's not a great technology for looking at these regions of the genome. So we created the CARD long-read sequencing initiative. And so the aim of this is to create an easy, accessible structural variant dataset for Alzheimer's disease or related dementias, also termed ADRD. So for this, we're going to long-read sequence around 4,000 human brain samples. And so with this data, we aim to be able to assess the role of structural variants in ADRD, resolve complicated regions such as the HLA, MAPT, and GBA locus. And what's really nice about the samples that we've picked is not only have they got matched short-read DNA sequencing, but a lot of the cohorts also have matched RNA-seq, so we can start to look at the functional effects of those variants. And finally, so the added benefit of Nanopore, obviously we can start to look at methylation differences between samples and disease. So we established the CARD long-read team, and so this is just a general idea of the leadership. Lots of familiar faces, I'm sure, and leaders in the long-read space. And we've all been working together on this issue of how do we create the most high-quality data set in an affordable and scalable manner. And we realized really early that there was three main challenges for creating such a data set. So first, we had to develop a wet lab protocol so that we could do nanopore sequencing at scale from thousands of human brain samples. Next, we needed a scalable computational pipeline to generate de novo assemblies, variant files, and methylation calls. And finally, we needed to have an approach for the data storage and access. So everyone here knows how big nanopore data can be. So per genome, it's about 1 or 2 terabytes. So if you times that by 4,000, it's a huge amount of data. So we've been very fortunate here to get support from nonprofit organizations to support us with this. So now I'm going to present the first part, which is our high-throughput wet lab protocol for long-read sequencing human brain samples. And so all of our protocols are put straight onto protocols.io. There's a QR code there if you want to go and look at our protocol for frontal cortex. And so with this protocol, you yield really high-quality, high molecular weight DNA, and you get N50s when you sequence the DNA of around 30 kb and around 30 coverage for the R9. And so we are actively still developing this, and a lot of the work is done by 3 great scientists in the lab, Lutz Malik, Bree Baker, and Meza Abdelhalim. And we're also running other projects, not just brain projects, so we've also developed very similar protocols for frozen human blood and for frozen cell pellets as well, and the QR codes are there. And I'll just give you a really brief overview of how that protocol works. So obviously, doing thousands of samples, we need to try and automate this process. So our DNA extraction is done on the Kingfisher Apex robot, and we use a PacBio nanobind tissue kit. We then shear the DNA to a target of 30 kb on a megaruptor 3, and the reason why we target the 30 kb is it's a nice balance between having reads that are long enough to do downstream de novo assembly, but then also maximizing the data output from a single flow cell so you can get 30x consistently. So next, we do the manual library prep with the LSK110. So we have automated this process now with the LSK114, but I'll mention more of that at the end. And finally, so we sequence the library for about 72 hours, and we usually do about 2 or 3 loads. And so this is what an average sequencing run looks like for the R9. So we've got a read length histogram, and you can see here we generally don't see any enrichment for short reads, and we've got a nice peak around 30 kb, and you can see by the MUX scan, so this sample will have had 3 loads. And so we're really fortunate at CARD, so we've got 2 P48s and a P24. And so we've been really gearing up, so how do we go from sequencing a couple of 100 samples in a year to a couple of 1,000 to more of a production scale? And through that work, we're now at a point where we can sequence quite comfortably about 200 samples a month. But obviously, one of the downsides to sequencing at that scale is it's not possible to do the base calling on the machine, especially because we use Guppy in super accuracy mode. So once we sequence our samples, we push the Fast5s off the sequencer, and then we've got a couple of options on where we can do the base calling. So we can do it on Google Cloud, and there's been a lot of efforts from our data science team to optimize that protocol. And so for the base calling now, it's about $130 per sample to do it on Google Cloud, which is about £100. And we've also got the option at NIH to do it on the NIH HPC server, which is called BioWolf. And the added benefit of that is we can do the samples for free, but we have to do it in small batches. So we've used the wet lab protocol, we've extracted our DNA, we've sequenced it on the R9, and then we've base called it. And now I'm going to run you through the scalable computational pipeline that we've generated that will give you de novo assemblies, variant files, and methylation calls. So this work has been led by Misha Kolmogorov at NCI and Benedict Payton at UCSC, and it's available as a preprint on bioRxiv, and the QR code's there. I'm just going to run you through how the workflow goes and also the benchmarking that we did with the human genome cell lines. So this is an overview of the Terra workflow. So you can access it either on a Terra platform or also on GitHub. And so the input is the unmapped BAM with the methylation tags, and then it can go down 2 routes. So either it can go through a reference-based approach, or it can go through an assembly-based approach. And so for the reference-based approach, so you can take the reads and you can align them either to HG38 or CHM13 with Minimap2, and then you can use Sniffles2 to call the structural variants, and Pepper Margin Deep Variants call the small variants. And then the output from this is a Sniffles VCF, a haplotype resolved methylation bed, and small variant calls. You can then go through the assembly-based approach. So it uses the tools Shasta and Hadoop to create a de novo phased assembly. And then from this de novo assembly, it uses a tool called hapdiff to call the structural variants, which outputs an assembly-based structural variant file. And then the final step, so it can have super accurate calls, it takes the reference-based small variants with the assembly-based structural variants, and the final output is a harmonized phase small variant and structural variant VCF. And so we wanted to benchmark this workflow. So we processed the three of these human genome cell lines with our wet lab protocol, and then we ran them through the Terra workflow. And here we are showing the number of false variants stratified by mappability. So in the green, we've got regions of the genome that are in that are in high mapability, and then in the orange we've got low mapability regions, so complicated regions of the genome. And so this is where we've compared Nanopore to Illumina in the same sample. And so overall here, what we're showing is that Nanopore has a reduced SNP error rate compared to Illumina, especially in those low mapability regions, which are shown in the orange. And so next, we wanted to look at structural variant calling. And so we compared the two callers in our pipeline, so Hadoop and Sniffles, with QSV and PacBio data and HiFiSM. So for the different 4 plots, we've got F1 score, recall, precision, and the genotype accuracy. And so the take-home message from this is that Hadoop, which is the assembly-based caller, has very similar F1 score to PacBio data for the same sample. And so this is just an example of how, with the pipeline, we can look at haplotype-specific methylation profiling. So one of the outputs from the Card-Terra workflow is haplotype-specific bed with the methylation profiling. And then you can take that information and put it into Modband tools and start to look at regions and see if they're differentially methylated. So this is the gene SNRPN, and it's known to be differentially methylated. And you can see here, so in the middle, we've got the haplotype frequency. And this is separated by haplotype. And on the bottom, we've got, these are the reads, and in the red, these are the methylated region, and in the blue, we've got the unmethylated region. So this is a nice example of how, with this data, we can start to differentiate and visualize differences with methylation. And so just to summarize the paper, so from this paper, we've shown that we can detect SNPs with an F1 score better than Illumina short read sequencing. Small indel calling remains difficult within homopolymers and tandem repeats, but is comparable to Illumina calls elsewhere. And we can discover structural variants with an F1 score comparable to state-of-the-art PacBio methods, but obviously with a lower cost and higher throughput. So next, I'm going to present the work that we've done where we've applied this framework to long-read sequence the first large-scale brain cohort. So we have sequenced a control cohort, which are from the North American Expression Brain Consortium, which is the NABEC. And so with this, we've sequenced over 200 frontal cortex samples. And so one of the main goals of this initiative is to get the data out quick and make it easy accessible. So in light of that, so the first 14 of the brain samples that were included in the methods paper, they're now on Anvil. If anybody did want to access them, so you have to go through dbGaP, and once you get granted access through dbGaP, use that same email to then go to Anvil and access the data. So we are pushing all 200 samples to Anvil and also to ADWB, which is part of the Alzheimer's Disease Data Initiative, but that process takes a couple of months. And so we only mapped all of the BAMs last week, so we've not been able to do a really comprehensive analysis, but we ran Sniffles, so we could share some results with you guys today. And so when we ran Sniffles with that data, we detected over 80,000 structural variants. And as you can see here, as expected, most of those were insertions and deletions. So next, we looked at allele frequency, and so it's shown in this plot on the x-axis. And so as expected as well, most of the structural variants in this population are rare. And so we also wanted to start to look at the functional effects of some of these variants. So Kensuke Daida in the lab did some eQTL analyses with those structural variant calls. And so for this data set, we've got the matched short-read Illumina RNA-seq. And so we can see here a nice example of a structural variant eQTL. So this is a 700 base pair deletion at the MAPT locus, which is a big Alzheimer's locus. And so this deletion is a QTL for the gene CANSL1, which is also at that locus. And so you can see by the violin plot, so this is where the genotype correlates with gene expression. And I was specifically interested in this one because it's a deletion of a human-specific SVA, which is a retrotransposon. And so on the right, we've got a samplot just showing how it validated. So you can see on the top, we've got a homozygous carrier of the deletion, and we've got a complete drop of coverage. And then below, we've got a heterozygous carrier of the deletion, which is a partial drop in coverage, and on the bottom we've got homozygous reference. And so finally, we've also interested at looking at cell type specific methylation in brain tissue. And this work was done by Pilar Alvarez-Jerez and Riley Genner in the lab. And so the brain tissue that we're using to do the long-read sequencing is very heterogeneous, and it has different neuronal cell types. However, we've got an in-house cell line, an iPSC cell line, that we've differentiated into different neuronal cell types, like the neuron and the microglia. And so this gives us a unique opportunity to start to look at methylation differences between cell types. And so this is an example of the gene TREM2, which is a big Alzheimer's gene. So on the top, we've got the sequencing data from the neuron, and on the bottom, we've got the sequencing data from the microglia. And so in the literature, it's known that TREM2 is not expressed in neurons, but it's a known marker gene in microglia. And you can see that quite nicely here. So at the top, we've got that flat line of hypermethylated. And on the bottom, we've got a dip where the TREM2 promoter is, where we've got an unmethylated site. And then what we can do with this knowledge is go back to that region in our bulk brain data, which is shown on the right, the NABEC sample. And then we can use the cluster option in Modbam tools, and it can start to pull out those signals, and we can clearly see the one signal that's coming from the neuron and the other signal that's coming from the microglia. And so what are the next steps for CARD? And so all the work I presented previous to this was on R9, and now we have completely moved over to R10. And we did do some benchmarking studies where we went back to the 3 human genome cell lines, we resequenced them with R10, and then did a comparison. And so overall, the R10, no surprise, did better on almost every benchmarking category, especially with reference indel calling. So outside of homopolymers and outside of tandem repeats, it's now in line with Illumina. So moving forward, we do everything with R10. And so now that we have moved on to the LSK14, we have been working with Jade Bartello and Oliver Holman at ONT to optimize this on the Hamilton NGS star. And so we've been optimizing this for about 10 months. And in the last couple of weeks, it's been brilliant. We've finally got to a point where we can library prep 48 samples in 4 hours on the robot, which is brilliant. And so you can see here by the N50s and the pass rate, we still need to do some more optimization specifically for the brain, but this is really moving in the right direction, and it's been a big win in terms of getting high quality, high throughput sequencing done at CARD. And so we're also interested in sequencing more diverse populations. So the first cohort that we sequenced, the NABEC, they were European, and they were used because they were our in-house cohort where we had lots of tissue available, and we also had lots of omics data sets to start to look at the functional impact. But we're just about to finish our second cohort, which is the HBCC cohort. And so this is a Black American cohort and our first disease cohort as well. And so we are open to sequencing other cohorts if anyone wants to collaborate and has a good interest in Alzheimer's disease and related dementias cohort. The only general requirement is that we prefer brain tissue, and also, so these need to be broadly consented to data sharing, and the samples need to be able to be stored and go through the dbGaP process. And so everything that I've presented today has been regarding germline variation, which is variation that's inherited. However, we're also really interested in looking at somatic variation, which is any genetic alteration that occurs after fertilization. And so somatic variation has already been associated with Alzheimer's disease and related dementias, and also with aging, and we know that it can have a functional impact in the human brain. But the only issue with studying somatic variation is you usually need really high-depth data, and so a lot of the previous studies have only focused on SNPs and have been using short read. And so when we were developing the wet lab protocol, we actually generated a huge amount of data for this one sample. And we ended up with about 400x frontal cortex data. And so we thought, hey, this is a great opportunity to have a really high-depth data set to start to look at somatic structural variants. So we generated high-depth data for a total of 4 regions. So we've got the frontal cortex, the parietal cortex, the visual cortex, and the cerebellum. And so with this, we aim to be able to utilize the data to better develop tooling to look at somatic and mosaic variants. We're hoping to be able to start to assess the presence and frequency of mosaic variants across different brain regions. And also the benefit of it being nanopores, we can also start to look at methylation differences across the brain. And for the analysis, we've been using Sniffles 2 in mosaic mode and working with Fritz Sedlacek and Lewis Powlin. And so Lewis is going to be giving a talk in the DAC cave later on, specifically on Sniffles, and that's going to be great, and I encourage everyone to go. So finally, this is just an example of where we've got the 4 different brain regions, we've ran Sniffles in the mosaic mode, and we've been able to successfully detect a low frequency variant just in one of the regions. And you can see here, we've got a 500 base pair deletion that is in the parietal cortex. And so just in summary, we've developed an efficient and scalable wet lab and computational protocol for nanopore long-read sequencing that seeks as a genuine alternative for short reads for large-scale genomic projects. We've now generated a new long-read sequencing resource for Alzheimer's disease and related dementias from about 200 brain samples. And there's lots of applications for this data, and we've shown that we can start to use this to look at somatic variation in the human brain. And so I just want to say a big thanks. You can see lots of names on here. This has been a huge collaborative effort, and I'm sure over the next couple of years there'll be more added. And I want to say a special thank you to our support at Nanopore. So we've had a lot of help from Andrew Markham, Jessica Anderson, Jade Bartello, and Oliver Holman. And I just want to say a massive thank you to Nanopore for inviting me here today.